Good morning to esteemed unit convener and my colleagues. Today I'll be presenting the article titled Quantitative Analysis of Post-Translational Modifications in Human Serum Transthyretin Associated with the Familial Amyloidotic Polyneuropathy by Targeted LC-MS and Intact Protein MS. The article has been published in 2015 in the Journal of Proteomics, and the study has been conducted by multiple researchers in Spain. Before directly moving into the context of the chosen article, I will present a brief overview of proteomics to highlight why I have chosen this particular article for presentation. The term proteomics refers to the characterization of the proteome in terms of protein expression, structure, function, structural modification, and interaction among proteins at any stage within the cells. Since proteins are the actual functional molecules, proteomics study provides a crucial information on the various cellular activities and pathways that occur in healthy and diseased cells at the molecular level. Among the different applications of proteomics techniques, the capability to study post-translational modification is extremely important since post-translational modifications increase proteomic diversity. For example, DNA usually encodes 20 amino acids. Protein contain more than 140 residues due to diverse post-translational modifications. Furthermore, these modifications play a key roles in functional proteomics, including various pathways. Since protein analysis is the primary way to identify post-translational modification, I have chosen this article that discusses the quantitative analysis of post-translational modifications in human serum transthyretin that is linked to familial amyloidotic polyneuropathy, or FAP. FAP actually is hereditary amyloidosis, which associated with amyloidogenic mutated transthyretin, apolipoprotein A1, and glysolin. This study particularly probes the post-translational modifications of TTR at cystin that are believed to play a crucial role in the onset and progression of amyloidosis. The research method that the author used, they actually used two key LC-MS based proteomics techniques, namely intact protein analysis and high resolution extracted ion chromatograms for their proteomic study. Firstly, they isolated wild-type transthyretin and amyloidogenic tetrameric protein isolated from the human serum samples, and expressed mutant human TTR in E. coli. Isolation of both TTR forms was performed via immunoprecipitation in solution using a polyclonal rabbit anti-human TTR antibody. Thereafter, the TTR was digested using different proteases such as trypsin, chymotrypsin, arginine, asparagine, and glutamate at 37 Celsius. The digested sample were analyzed using QTOF spectrometer coupled to nano HPLC system to select the most suitable enzyme for the study. The immunoprecipitated wild-type and mutant TTRs were firstly digested using arginine for analyzing intern peptides with modification at cystin and trypsin proteases. The digested sample were then analyzed via targeted LC-MS high-resolution extracted ion chromatogram strategy. The authors performed intact protein analysis in which immunoprecipitated wild-type and mutant TTRs were analyzed using ultra-high resolution QTOF. Measurements were performed in the range of from 50 to 1500 mass to charge value, and in the charge envelope positive 14. Intact protein analysis basically analyzes the protein without enzymatic digestion or fragmentation and helps to characterize post-translational modifications, sequence polymorphism, and cleavage sites. Simultaneously, the immunoprecipitated TTRs have been studied using top-down analysis, and this method basically involves fragmentation of the protein to obtain mass spectrum with the mass to charge values of the fragments, and fragments of interest are further investigated by fragmenting through MSMS technique using collision-induced dissociation or electron capture dissociation. In this presentation, I will discuss the key findings presented in the article. The authors aimed in this study to identify post-translational modifications of TTR at cysteine, and the commonly reported modifications are usually S-sulfonation, S-glycine-cysteinylation, S-cysteinylation, and S-glutathionylation. However, intact protein analysis detected signals with the mass to charge values corresponding to the different cysteine modified form for both wild type and mutant TTR. The table provided below shows a good agreement between the calculated and experimentally obtained mass to charge values. And to confirm the fragment structures obtained from the intact protein analysis, a top-down approach was used for the wild-type and mutant TTRs. For example, the 996.5 mass to charge was correspond to the S-cysteine glycine modification at cysteine. Top-down approach helped to identify structures of the fragments corresponding to the different post-translational modifications of TTR at cysteine. However, the result of top-down approach and the intact protein analysis were found to be in agreement. Based on intact protein analysis and targeted LCMS high-resolution extracted ion chromatogram approach, the study comparatively evaluated the proportion of different cysteine forms of post-translational modifications at cysteine of TTR. From the intact protein analysis, free cysteine, S-cysteine glycine, and glutathione fractions are found to be more abundant in both wild-type and mutant TTR compared to the high-resolution extracted ion chromatogram. What's interesting is the intact protein analysis shows both forms of TTR are almost equal in concentration. However, absolute quantification via high-resolution extracted ion chromatogram approach shows that wild-type TTR is much less in abundance than its mutant form. This sums up the key results reported in the article. Anyhow, the study has aspects that can be appreciated, and at the same time it has certain limitations. First, the authors have carefully selected the enzyme for TTR digestion used in the targeted LCMS high-resolution extracted ion approach based on the LCMS results obtained from the enzymatic digestion of TTR using different proteases. Among the different enzymes tested, the arginine was chosen for analyzing N-term peptide with the modification at cysteine since it provided the desired fragments and MS signal intensities. Second, the structures of the peptide fragments obtained from the intact protein analysis were verified using top-down analysis for both wild-type and mutant TTR. Third, the results obtained from intact protein analysis of wild-type and mutant TTRs were compared with those obtained from the targeted LCMS high-resolution extracted ion study. Fourth, multiple samples of wild-type and mutant TTR have been used in the study, which improves the quality and robustness of the results. Fifth, the article is well written and the research methodology is clearly described, providing sufficient details so that other researchers can use the methods while performing similar studies. Sixth, the authors clearly explained the discrepancy in the quantitative results. For example, the percentage of the modification present in the TTRs obtained from intact protein and targeted LC-MS high-resolution extracted ion strategy. However, according to the authors, intact protein analysis is capable of providing relative quantities of post-translational modifications present, whereas the targeted LCMS high-resolution extracted ion approach is quantitative method with higher sensitivity. And as I said before, the study has certain limitations. The intact protein analysis identified the S-sulfur modification. However, the relative percentage of this form cysteine was not compared with the result obtained from the targeted LCMS high-resolution extracted ion approach. Furthermore, intact protein analysis found one unknown fragment in wild-type TTR and another in the mutant form. However, the authors did not attempt to identify this fragment or the corresponding post-translational modification of TTR. The result obtained from both intact protein and targeted LCMS high-resolution extracted ion approach were not compared with literature values, and neither do the authors mention whether or not there are literature reports to support their findings. In conclusion, familial amyloidotic polyneuropathy is a fatal hereditary amyloidosis condition, and post-translational modifications of TTR at cysteine are believed to play a crucial role in the onset and progression of this disease. The study probes the post-translational modification at cysteine of both wild-type and mutant TTRs isolated from human serum samples using two key proteomic techniques, the intact protein analysis and targeted LCMS high-resolution extracted ion strategy. Qualitatively, the results obtained from both analyses are in agreement. The study has certain strengths as well as limitations that have been discussed in the previous slide. In conclusion, the authors could have used a bottom-up approach for their study. This approach allows the quantitative detection and characterization of proteins using affinity tags and stable isotope labels. The references used for the presentation are listed here. And here some additional references that can provide insight into the current advancement and state of art in proteomics. And this will bring us to the end of the presentation. Many thanks for your kind attention.